All right, welcome back. Here we are. We're now moving on to chapter 11. And aren't we speedy? We're just moving right along here. And, I told most folks that we need to strap on the seat belts because it's going to be moving fast. Well, that is true, and we're doing the best we can. So, chapter 11 covers one of my favorite topics is the interactions between microbes and humans. And then this is a perspective that I have that microbes are not set as nasty things that they scheme at night to figure out ways to kill you. And they're just growing in a niche, and if you open up that niche to them, they'll grow in it. And some folks do things that are not probably good health and or engage in activities that are not good, and then they know it's not good, but it gives an opportunity for some microorganisms to gain entry and that sort of thing. We're going to talk about that. And the reason I moved 11 to where it is because this is the key part of all the things that we're going to learn about the microbes. And I think, and I've done this is the third year I've done it this way. The students like it because it's why we do and study the things that we need to study. If you understand how the microorganism, what they're doing to form a disease state, then you're going to feel more inclined to want to study about how it's doing it. And so that's why I kind of move the chapters around so that you get that perspective. And again, it's all part of the first week. I want to reemphasize, I've been getting questions. And, as I'm formulating class, I do want you to know that when I have due dates and things, those are suggested due dates. And I'll make sure that the way the forms are, it doesn't allow me to do that. But I just want you to know that. I know that everyone's busy and you're doing the best you can. And that's the way I assume. And so if you're a few days late, that's okay. I'm not going to take points off. I want you to do the work. And the idea is I want you to learn the material. And so we're doing whatever it takes to do that. And so I just want you to know that's where we're at. We'll be doing week 2 before long. So let's get done with chapter 11. And I do it in sections so you can kind of take a breath and see it. I hope that's working out for you. So we're going to look at how we differentiate against, again, these courses, a lot of them are vocabulary. And we have to understand the vocabulary. And that's why I use the Latin terms to help because I want you to systematically learn. And so colonization, infection, and disease is really what we're going to talk about. And that's why when we start to go back and learn more about the bacteria, now you'll see why and how they apply these certain things to do what they do. Enumerate the sites where normal biota is found, and this is a really cool topic. One of my favorite topics is the Human Microbiome project. I'm really into understanding how microbes organize. One of my professors at State, he was one that's published a lot of papers about where microorganisms are, like under your armpits or your feet or various places on your nose and face and various things like that. And it changes during the day because our pH changes, our body temperature changes, and all sorts of things. And I find that fascinating. I don't know, I guess I'm weird, but the microbiome is really, really important, and we have to understand it because there's one thing I have observed, and I want to share this, is probiotics, and I've mentioned that before, but what they did was there was this huge study for what is the normal flora, what is the microbiota in babies when they're born, and what organisms are predominant at that time. And they did a worldwide study, and it came down to about 12 organisms. And so this one company that I'm aware of that does probiotics, they used the 12 organisms that were the common ones. And I thought that was really, really smart. And I personally take probiotic of that type. But the idea behind that is there's certain things that we need, and microbes, they're not always harmful. There's a group of bacteria that are helpful. So we're going to talk about that and trying to set us up. So the human microbiome is really the sum total of all microbes found on and in a normal human. Now, they've been using new techniques called PCR. I think we just mentioned it in the previous chapter. Polymerase chain reaction and DNA barcoding. And I'm not worrying about you knowing all that stuff, but the idea is that they can detect very small amounts. And what it's done is absolutely blown apart the concept that urine is sterile. Now they find transient infections where bacteria are colonizing. And things CNS, well, it's not as sterile as we thought it was, and that sorts of things. Our bodies and immune systems are constantly fighting back and forth, and I just want to confirm the notion that the microbes are constantly being presented, and we push back and we do those sorts of things. So the human microbiome is really critically important for our health and well-being. And if we have a disruption, and that could be as simple as gargling with a mouthwash, and it changes the population of bacteria. They're finding, let's say, gum disease seems to be a higher coincidence with patients that have the particular diseases as they get older. So there are correlations with disease and not having certain types of bacteria. The disease I was referring to is Alzheimer's. And if gum disease and Alzheimer's, they're thinking there might be some kind of link. The idea, though, is that having that microbiome is really, really important. For the most part, resident microbiota colonize for long term and don't cause the disease. They're there, and let's talk about it in our gut and having bacteria that kind of the good ones that are sitting there. That's like probiotics, and it's like a parking lot, and there's a lot of good people, really wonderful people, and we want them to park there because we want good people around. And so just the fact that they're parked there means that folks that are not so nice, undesirable bandits or whatever, we don't want to have a place for them to park. And so the more good ones that we have, the less probability of bad ones. You see what I'm saying? And so there is that give and take. And so if we partake in a particular activity that diminishes the healthy ones, then you can see the bad ones will kind of move in. So that's kind of the long-term aspect of what they're referring to here. Infection, microbes get past host defenses and enter tissues and multiply. So this is where either we have let our guard down or something, and something now invades, and that's a real problem. So that's an infection. Disease is deviation from health. It's a pathologic state that results in cumulative effects of infection, damage, or disrupt tissues. So the disease state is deviating. Of course, it's not a normal state. And then an organism is producing something. Now, it could be as harmless as a waste product, but it could be hugely toxic to us. So a good example would be the microbes in our colon. And you're a truck driver and you don't use the bathroom as frequently as you perhaps should. These microbes are eliminating waste and things, and a lot of them build up and are present to tissues that are there, and you might see a higher incident rate of colon cancer because these chemicals that normally shouldn't be there or should be eliminated, and it limits the exposure, tend to be there longer. So that's just a weird way, I guess, of trying to talk about disease. Infectious disease is a different deal. A pathogenic state caused by direct microorganisms and their products. It could be that we're talking about bacteria that you may get associated with high numbers of certain organisms. There was a study, how many organisms does it take to start a particular type of disease? And it varies on the types of organisms. Like tuberculosis doesn't take many at all, versus having, let's say, food poisoning. Depending on what it is, you may not have any organisms at all. It could be just the byproduct of toxins that can make you sick, like staph, and they just excreted all their waste products on this potato salad and you eat it, and the toxins alone were enough to cause the really bad part. But that's not really infectious. That is a result of food being infected, I guess. But infectious disease is more of having an active organism growing, causing problems like tuberculosis or something like that. Now, I'll show you a picture here. And what we have is colonization of these microbes in a particular orifice or something. I want you to think about that. These grow, and some of them can double every 20 minutes, and so it doesn't take long. So you can imagine if you have a wound or if your immune system is weakened, and an orifice of some kind might be more colonized, and it runs to a higher probability of an infection. And so again, this could be bacteria starting up, forming a biofilm, and biofilms are going to be something we're going to be talking about. But it's organisms that are binding, and what happens is it enables other microorganisms to start binding, and once they're kind of working together, it starts to damage, and we start to see a tooth getting infected, causing occlusal or a tooth decay of some kind to a toothache, and makes a dentist rich and that sort of thing. So the Human Microbiome Project is a worldwide effort that happened around 2008, I think that's right. Colonized microbes living on human bodies when healthy. We'd like to know that. And then, how does this shift during diseases? What's going on? What predominates and that sort of thing? And then we used powerful techniques that we've learned in genomic sequencing and the PCR and DNA barcoding and all those sorts of things, and we start to correlate this type of data. Now, I did it the old-fashioned way using biochemical tests, and took forever. And now the types, this gentleman here is working. You can see he has a flame. He's got his gloves on. He's practicing really good aseptic technique here, by the way. If you notice, the camera shot is an interesting one because it's in a hood, and it seems to be under a protected top. And he's created a pretty good aseptic area. And he's got some plates here. Looks like they're going to be a diagnostic type of plate. And he's got his samples, and it looks like DNA, and it looks like he's got a hockey stick going on there, spreading bacteria on a plate. And it's very common. It is to sample lots of microorganisms from various things, and then you identify them by growing them. Well, genetically, we can do this without even growing them. We just check for the presence of their genetic material, and that's usually enough to let us know what's going on. And that's a whole new realm of what's going on. So some of the results that we've seen: human cells have about 21,000 protein encoded genes. Microbiota has 8 million. Now, one of the friends of mine, who's a pretty famous fella, he likes to think of our bacteria as the extended genome. And I think that's a really cool term. That it is. So we have 8 million protein encoding genes in these bacteria. So they're complementing, they're adding to the repertoire of our exposure to stuff. And a lot of these things could be very important. Microbes are found in locations that we thought were sterile. I've made reference to that already. 100 million viruses per gram of human feces. Pretty scary. That's a lot of viruses. 100 million. Well, actually, it's 100 million viruses. It's really not that much, but it's okay. All healthy people seem to harbor potentially dangerous pathogens in low numbers. We carry them around. I'll guarantee you right now I can find Staph aureus somewhere on your face and around your nose, your ears, or something like that. Makeup of one's intestinal biota can influence overall health. I totally buy into that. I think that's totally true. Sites with microbiota, sites previously known to harbor normal skin, mucous membranes, upper respiratory, gastrointestinal tract, including the mouth, outer portions of the urethra, external genitalia, vagina, the external ear, external eyes, all of this. We're just loaded with various things. and that's a good thing. It's part of who we are. Um, Additional sites now thought to harbor at least some normal microbiota: lungs, lower respiratory, bladder, breast and breast milk, amniotic fluid in the fetus, of course. So those are important things. Sites in which DNA from microbiota have been detected. Here we go: the brain and the blood stream, and those were always considered sterile. The CNS and that sort of thing is what they're referring to for the brain. Pretty interesting. So the microbiota are a mixture of various different types of microorganisms found pretty much everywhere at any one point in time. Acquiring the microbiota, the human body has a variety of environmental niches. There could be pH, temperature changes, nutrients. You could be stressed out and pH will change, and maybe the microbiota will change as a result, and that changes yet even more of the surface of the skin. And now we see that we have all sorts of problems. So anyhow, uh, you can get the idea. Microbial antagonism. Normal biota are unlikely to be displaced by incoming microbes. That was the parking lot analogy I gave you. If good people are parking, then the bad ones can't park. There's no place to park. And there are a finite, limited number of attachment sites or parking places. And so that's a good thing if you're living healthy and doing that. Chemical or physiological environments created by resident microbiota also set up this hostile environment. One of the things I worked on with a probiotic was there's a certain lactobacillus that produces a substance that's really toxic. We found out that it's like the number 2 of the most toxic substance list, and it turns out to be a lactobacillus reuteri, and it produces a chemical that's really, really very potent, and it prevents other microbes. They can't develop a resistance to it, but the microbe that delivers it seems not to be as affected because it's kind of like at arm's length. It's really interesting, but I just throw that out there for you to think about. These things are dynamic. So factors that weaken the host defenses and give us more susceptibility is age. You think about how the tonus of the skin and various things like that changes. Extreme youth, so you have an immature immune system, something like that. Genetic defects in immunity and acquired defects in immunity, like AIDS. These viruses, you don't have AIDS when you get HIV initially. You have an HIV viral load, and it mutates. It's a RNA virus, and it picks up a mutation. Has reverse transcriptase, which takes it to DNA, which is sort of an exception. But the idea behind that is that it has to mutate now in order to infect a particular type of immune cell. And when it does that, it takes out the helper T cells, and boom, you are essentially, your immune system is like your shields are down, and you can't respond to a particular infection, and that's not good. Surgery and organ transplants. Obviously, they give you immunosuppressants when they do organ transplants, and surgeries are opening your body up to potential infections, or having a device put in or something that may harbor something. Chemotherapy, immunosuppressant drugs for other reasons. Physical and mental stress. You would not believe how much mental stress can really have a negative impact. Pregnancy, and then of course other infections going on all at the same time. So you think of it, it is being attacked from all sides, and our immune system does an amazing job to keep all this in check. And in addition, our immune system also detects cells that turn cancerous. So I want you to think about the task that our immune system has. It's not only threats from outside, it's threats from within inside too. So it's quite a task. The colonization of the fetus. Until recently, the uterus and contents were thought to be sterile, and now we're finding out now this is not the case. We have a diversity of microorganisms. Intestines were colonized in utero before they come to the world, and that's why I wanted to bring that out to you, because it's important to know. Lactobacillus provides baby with necessary enzymes to start digesting the mother's milk and just in overall good health. Other species protect the baby from skin disorders and other conditions. That's good. Milk contains about 600 species of bacteria and sugars that babies can't digest, but the bacteria can break down these particular sugars. And so it's all part of the systematic thread of life, I guess. Everything kind of is related to another. So, where the babies get their microbiome? Well, obviously the mother, the environment, various things, caregivers, all sorts of things. And we're going to talk about more about how we respond to these. But I'll leave us with this slide, and we're done with the first section of chapter 11. And what an interesting topic to contemplate if we think about the environment in which we live. And it's not so much worrying about the bad ones, is also making sure that we're associated with good bacteria, and that's a good thing. So next time we'll move on to the second section here, and which really gets me going, and it's going to take a while, but it's very exciting, and I will see you when that time comes. Have a good one.